memorization. You just have to remember this. So sigma factor in bacteria is the one actually constant that recognize the initiation part, promoter part. GU is the wobble base pair in tRNA, and codon is anticodon, and codon that's the wobble base pairing. Then, well, those are in the 60%. Then it jumps to 90%, and those are easy expressions. So, yeah, so overall it's pretty good answer. But that said, so I think just to make you spend more time to practice, I'm going to scramble all the questions and reshuffle the answers. That way, at least you do some practical run just to prepare yourself for exam. Instead of giving all the questions exactly the same. Are we okay with it?
Would you say the same thing?
I'm going to scramble all the questions and answers from now on. Run permutation. Right, yeah.
Okay.
So that way it will be a little more difficult if you sit next week and say this one is the at least you discuss what the answer is. So let's finish chapter 6. We finished the Spelman, finished the double helix, you see, right? Okay, here's the question. E. coli genome is about 5 million base pair, and its average protein is 13 amino acid. And now, let's do a clicker. Oh, sorry. Just a minute. I forgot I haven't even set up the Is this open? Yeah, okay, good. Yes. So the entire genome have a 5M with 5 million base pair. The average protein length is 300 amino acid in length. Okay, how many genes are correct? Genome. Okay, that's good enough. Let's see what's it's not showing here. Okay, so okay, C. Why C? Yeah, C is a majority. D is also chosen. B, someone also choose B. Okay, let me show you what the actual E. coli genome is. Now the funny thing about the bacterial genome is you actually don't really have a very good number. In human genome, remember last time we were not sure because we So let me show you what the E. coli genome actually look like. This is a, oh, this is the number is very small. I need to, how do I adjust the size of this? Okay, that seems to be a bigger enough. So this is a genome called E. coli strain K12, and I have the sequence in a GenBank format. So this is basically the E. coli genome. It's the E. coli strain K12. Entire genome is basically like then if I ask you how many genes in E. coli genome, this is that E. coli is 5 billion, and imagine a human genome within the billions. So basically, it's hard for us to actually even to know how many genes in human genome. E. coli is easier because there's no intron, so we can actually find out more easily what gene is. So in E. coli, we actually know how many genes. Well, at least we have a better idea. So those are the number of proteins in E. coli, protein sequences. So each, you see this greater than sign, that means there is a new protein there. And the ends here, so another greater than sign, that means another protein. Those are called FASTA format. So how do we know the average length of E. coli protein is 300? We just count how many, this one, this entire row is probably 80 or 70 characters. So 1, 2, 3, 4, 5, 6, 7, 83, 20. This one is about 350, let's say. And we count another one. This one seems to be 1, 2, 3, 4, 5. Oh, 40. Sorry, I counted wrong. 1, 2, 3, 4, 5, 6, 7. 7 times this is about 600. So, and then we just count all the proteins that average. The average seems to be about 300 amino acids. But each amino acid is coded by 3 nucleotides. So one amino acid is 3 base pairs. So we have an average length is 300 amino acids, and give about 900 base pairs. But usually there are promoter regions and other non-coding parts. So let's give another 100 something. So 5 million divided by about 1,000. That's roughly about 5,000 genes. The actual number, the actual gene number is actually in this one is actually 5,287. Yeah, so I guess it's actually in between. Yeah. So, but I guess 5,000, usually most textbooks will say around 5,000. 1,500 is also a close enough. I guess I would say a little ambiguous, just to make fun of. Okay, but this is actually not a fixed number. In fact, different strain actually shrink and expanded. So this number can change to sometime around 500, sometimes actually be closer to 6,000. I have seen some of the E. coli genome can even have 6,000 genes, closer to 6,000. It's because the bacterial genome are more plastic or fluid. They actually can take genes and throw away genes more easily than eukaryotic like us. So it looks like
So would you ask us to add that 100 to the 900 base pairs if you access to some like a test or something? Because when I just divided 5 million.
Oh, if you just divide it by 900.
It gives me 5,500.
Oh,
Like
yeah. Yeah, I guess in the exam, I probably good to give you a more clear instruction. I wouldn't give an ambiguous.
Right.
Yeah, unless it's a small essay, I actually don't explain the answer. But, okay, so that's the exercise. So what I'm, this is some simple bioinformatics work I'm doing in here. We may do some simple exercise just for the lab. It's actually very useful if you at least know something of this. It's very straightforward. It's much easier than experimental work because once you know how to do it, it always works. It's not like a stripping colony. Sometimes contaminate, sometimes. So, if you know how to do the informatics, it's always work. That's a good thing. Okay, so. Let's do genetic information. Oh, that's a plasmid one. Plasmid usually confer the accessories optional when people buy car, they're optional. That's what we consider the plasmid activity. So I don't think I will go over all this, but okay, for DNA replication. We know, you all know it's semi-conservative. Now here's another question. Let's do a pulling on this one, see how it goes. And let's first read the question. So, there's a culture of bacteria. Let's put in a flask. It's actively growing. Let's say it's in log phase. And then we add radioactive cytosine. They say it's C. And it's radioactive. Add this into the culture. And after cell has replicated once, what does that mean? It goes through cell cycle once. So one cell split into two cells. And then we have 5 choices. Let me put it here. So. One of the daughter cells, but not the other, would have radioactive DNA. Either of the 2 daughter cells. Oh, I see. So the question does not distinguish mother cell and the daughter cell. So it basically says there's one cell is split into 2, and it's calling both of these 2 as daughter cells. So it's, I guess, the person who wrote this don't think bacteria age. So okay. Neither of the 2 daughter cells would have radioactive DNA. Both daughter cells will have radioactive cytosine paired with non-radioactive adenine DNA replication. So basically say that C will pair with A. DNA replication will not occur because 2 radioactive bases not proper base pairing. So we can just eliminate the wrong ones. So this one will be gone. This one will be gone. Now look at A, B, C. We know the DNA replication is semi-conservative. So if I have the old DNA strain, it's going to set when this is in a, oh, maybe I should draw it here. So in a That's by in the first generation. After that, it's going to separate, right? And then the new cell going to have a this is not good. Right. So now I know the old one is radioactive. So this, because of the cytosine, so they're going to be radioactive cytosine there. So this is hot DNA strand. So in the new cell, all those blue should have active one. So that's basically what it is. So A will be gone, B will be gone, so we have just C left. So let's see. Oh, excellent. So most of you should see already. So this new PowerPoint always put this one in there. Yeah, so it's good. Why don't you take a piece of paper, write this question, describe this in a piece of paper, write your name and send it. And basically the question is, when I say write a brief explanation. Say for radioactively labeled DNA in chloride cell. If cells start with radioactive labeled DNA after one generation, because of semi-conservative DNA replication, most of the cells should contain a copy of the radioactive labeled DNA. Yeah, basically write the explanation why C is correct. Yeah. Instead of write C. I mean, you can look at this figure. That's basically tells you. Yeah, just listen to the I just leave it thank you. No, I just need a name. Okay, so for the DNA polymerase activity, most of you answered the question correct. So, but here is the exercise. Again, okay, so this is a question on DNA replication. Although it's basically the question actually talks about PCR already, but this question itself is about DNA replication. So on the top is the DNA template, at the bottom is the primer. And then the question asks the DNA tag polymerase, which is going to replicate the template DNA, and the question is, which sequence is actually will be synthesized by the DNA polymerase?
I mean, you're asking for the rest of the sequence.
No, basically, maybe that can. Let me try to trick again. So, okay. So basically, this is the primer. The bottom is the primer. The top is the template space. So the polymerase need the primer to work. It doesn't synthesize just out of nothing. So it needs something to add on. So the question is, given that primer, the template, and the DNA polymerase is going to synthesize the rest of the sequence, which sequence will be synthesized? You can discuss among each other how this is going to work out. Then let me put the polling again. Oh, someone already input the response. Okay. Sorry, it doesn't read very well. I'm going to put this up. Okay, so okay. Either you should have a discussion, you should just stare at it. I don't see a response. You just look at the screen. No worries. Why? You are the first one input the answer. Why do you explain? Well.
I don't know if it's right, but you said this was a primer, and this is the primer, right?
Right.
And then you want to synthesize it with the tag polymerase, and so this is 5 to 3 prime. I did this from 5 to 3, but it will go this way starting at 5, so it'll be C, T, 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 A, C, and I think it's this one.
Okay, good. Yeah.
Right.
Everybody agree? Oh yeah. So it should be C. Let's see. Oh, let me see. Yeah. I guess we are already said C. But okay, so this bit, I guess if I give you more this kind of exercise, also prepare you for the exam, I guess. So, okay, so DNA polymerase, DNA polymerase 3. Then again, those are things you have to remember, I guess. So you need, we need a primer to actually, and for DNA polymerase, the primer is actually the RNA in the cell. Right? For PCR, it's DNA, but in the cell, it's actually RNA. Right? Okay, replication form, DNA helicus, just to unwind the double helix. This is the fun part. To be honest, with all these cartoons, I don't remember which one is which. So, but this is just good for review. Let's see. Okay, so the orange is the RNA primer. So the green should be the strand replicated. That's okay. The bottom is the leading strand. The top should be the lagging strand. Oh, I guess next one, the pink thing is DNA polymerase. Because single strand DNA. Okay, the ligand strand finally come out. So, what is this one? What is the big blue thing? Someone is a voice opinion. This is almost like a snow plow. The very front has to pull things apart. That would be. Helicase. Yeah, this should be helicase. Let's see. Yeah, okay, good. So, it's a cartoon. I mean, help us to understand the process. Okay. Bidirectional. If you think about, you can actually calculate how long it's going to take for DNA for E. coli genome to be synthesized. So here, DNA polymerase 3 is going to work synthesize 1,000 nucleotides per second. And how long is the E. coli genome? 5 million. So it's 5 million. And each second is 1,000. And so we divide by that times 1 second. That's how long it's going to take. If you just one enzyme working all the way. And in reality, there are two strands, but both strands are synthesized, and let's assume they are at the same time. So, but this will be, how long is that? 5,000 seconds. Yeah, 5,000 seconds, how long is that? How many minutes is that? How many minutes? 5,000 seconds, how many minutes? We divide by 60, so that should be 50 minutes. Wow. Actually, how long does E. coli to finish this? E. coli finishes one generation in about 10 minutes. So this is too long. What? This is oh, I'm sorry. I was actually, this is 83?
Yeah.
Thank you. I was actually divided by 100. Thank you. So this is just not right. So E. coli actually does almost 8 times faster. In fact, it should be 20 times because it's the entire generation is 10 minutes. That means its DNA replication finished much faster. So this is not right. How can E. coli do this? If the DNA polymerase work very slow, but in reality work much faster. How can they do this? Yeah, it's basically there are multiple replications to start, and then in the end you have to like it. So there are multiple. So like your computer, you have one. I remember in the old days when I have a phone, then say we have a dual core. Now we have a quarter core. So that's basically how we speed up your phone or computer. We have one CPU, now I have 2, I have 4. So the E. coli, if one enzyme finishes in 83 minutes, let's just, if E. coli can do it in 1 minute, that means there should be at least 80 different enzymes are working at the same time. So, okay, that's actually a pretty rough estimate. So, to give you some idea. Okay, so replication is also a property called proofreading. How can DNA proofread this? Oops. I'm supposed to have you an enzyme. I'm supposed to give you a question, but I forgot to put it on the application. Okay, let me write it here. So that in PCR, actually the mutation rate is much higher. So the question will be, the Taq polymerase PCR lacks which activity? 5' to 3' exonuclease. I guess it should be A, B, 3' to 5' exo. Nucleotide I guess this would be a multiple choice question. Basically, I just let me just say all of the above. just to it's obviously wrong just to make it a regular exercise, I guess. And then let me see what I can put in this. So that's a question. The question basically say in PCR the mutation rate is very high. The question is why the mutation rate is very high. This is probably part of the view of it. But, let me look at it from this side. I guess it's good enough. You can see them. So what is so the DNA? Well, the DNA polymerase works from 5' to 3'. If it had to proofread, it had to backtrack it. So it has to go from the polymerase works from 5' to 3'. If you want to, if it makes a mistake, it has to go back. So it cannot be in the same direction. It has to be in an opposite direction, 3' to 5'. And it cannot be synthesis. It has to be exonuclease removal nucleotide. So you have, only this one makes logical sense. So it should be. Yeah, so but it should be B. Yes. So I really shouldn't close this, but so kind of okay. So then the cartoon data. So now this is how the basically the principle of PCR now. It looks like a linear, but if I plot in a different scale, it shouldn't be a linear. This is PCR goes up in an exponential fashion. So this is linear because the y-axis is log scale. So that's the. Sometimes people forget about this. So actually, I probably put two different figures in, or 2 or 3 figures in the exam. Ask you to help. If I change the scale, it's not always linear. If it is a normal scale, it will be goes up like exponential, which means very sharp. Okay. So the basic idea of PCR is basically iteration and recursion. It's a computational thinking thing. Let me maybe I can go back to this later on. So in a lab. So this question is also about PCR reaction. So which are the following is true about PCR? This should be an easy question. Enable a small amount of DNA to be amplified. Addition poly-A. Addition poly-A only occurs in eukaryotic mRNA post-transcription process. So That's the eukaryotic transcription. Cut DNA into small fragments, no, that's restriction enzyme. Separate DNA from the nucleotide, no, that's gel filtration or agarose electrophoresis. Require RNA. No, inside of cell it has RNA fiber, but in polymerase reaction we provide fiber in DNA. So A is correct. It's still percentage C. Oops, I closed it again. I really should stop doing this. Okay, so here's part. This is the part where most of you, half of you, get it right. So in bacteria, the promoter is recognized by sigma factor, and how the transcription stops is by a terminator. So I think I had an animation. Yeah. But before the animation, the promoter usually have two regions. One is called Pribinol box, -10. The other one is called -35 region. This is the two box. Yeah, Pribinol box and -35 box. The termination will be have intrinsic, which is usually require the RNA secondary structure of something called row-dependent termination. Okay, let me show the animation. So that's this is sigma factor. The sigma factor bind to the promoter region and bring the recruit the polymerase to the DNA strain to the promoter region. So sigma factor binds to the Pribinol box, which is equivalent to the kappa box in the eukaryotic system, and then also the -35. The next part will be elongation. Yes. And then the sigma factor will be released and it move to elongation will move to the middle. Then you move to the termination part. Termination sequences. This one going to, what is this? This will give you a stem loop structure, basically a cross. So we have a stem loop form now, and then it terminates. So we probably need to do a summary figure later on. So okay, so and in bacteria, most of the genes are transcribed in operons. That's actually we will get to go back to the operon later on. Okay. For translation, there's a similar signal equivalent promoter. That's called Shan-Dagano sequences, and the translated part on DNA or mRNA called open reading frame. So this is actually the good question for exam. What I look? No, I don't know. So the G wobble basically is actually also the question most people, half of the class got it wrong, at least initially. So because it's tell you the answer, so eventually you get it right. The first attempt, like half of them are wrong. So So the GU wobble pair, although doesn't make sense in DNA or GT, equivalent to GT in DNA, but in the anticodon and codon base pairing, it actually is an advantage to have this because of the usually four codon, usually multiple codon codes for one amino acid, but not then altogether there should be 64 combinations, but not all of them actually have the majority of tRNA. Many of the codons actually recognized by the same tRNA because of this wobble position. So I used to work in a lab. Half of the lab work on tRNA structures. Look at this. Yeah. So, tRNA actually is one of the most stable RNA structures. Usually the mRNA is very unstable, and if you this is because in our body, if we see that RNA independent RNA unit, that usually is a virus. So our body have a lot of RNAs to cut RNA structure. So if you work on the mRNA to a northern blot, you don't want to spit into your samples. That will just kill everything. But tRNA is one of those things that is extremely stable. I was actually very surprised when I first working in a lab that people just put RNA everywhere. But it turned out this system is very stable, mRNA. So translation process. Translation is probably the most ubiquitous genetic and biochemical process. The machinery is so old, it's actually conserved from E. coli to, E. coli, archaea, and eukaryote, they're all the same. The mechanics, they're the same. And although the component will change, but they all have the same thing. So I also have animation. This is a very long lecture. If you think about this chapter is covered from DNA replication, transcription, translation, coding. What is BIO233? BIO233 is called cell biology or 110 I know is a diversity. 115 is something called form organism. What is 120 though? So does 120 cover it? Yeah, but they spend probably several weeks on this. Okay. Oh no, this is transcription again. Let me do a translation. Okay, this is the translation part. So translation has a clear signal, green light. This is a very good traffic light called start codon for translation to occur. Actually, not many biological processes have such a clear signal, but this is good. Those are IF factor, initiation factors. Start codon and ribosomal. This must be it's going to recognize the short-diagonal site. Okay, so we first. Okay, that's right. Yeah. Short-diagonal site to recognize. Also by base pairing. And then you're going to bring the initiation tRNA. So it's AUG, and the tRNA should goes from base pairing always is anti-parallel. So the other one should be CAU. then bring the large subunit, all the initiation factor will be released. Now it's the elongation part. And well, now, so this is now at the so-called P side, and there are 3 sides for this whole thing to work. A side, P side, E side. Basically, almost like a assembly line. Elongation, the green one, then move on to the next one, and then and it's almost like a train moving. There's a transfer formation of peptide bond. Translation use GTP. So moving along, then release one, come back next one. It's basically a repetitive process. And it's also a clear signal to stop here from the stop codon. UAG, the stop codon. Stop. Okay, so I guess we don't have much time. I can just summarize this. There's not much left, so we only have protein structure left. So let's summarize this whole thing. So we have DNA and RNA and protein. And the DNA going to this process will be right. This is the replication process. And this process is transcription. And this process will be translation. I guess it's called translation because the nucleotide are still considered one language. Amino acid is considered a different language. So translation here. Now, okay, now that's the fun part. So what's the start, run, and the stop signal in those processes? For replication, what's the stop signal? We need replication origin. Replication origin. Also, what's the, well, let's just look. For transcription, we need what? The start signal will be, we're all talking about bacteria here. For transcription, the start signal is what? Promoter region. So this is promote. So for transcription, we need promoter region. There's a -10 Pribinov blocks. -35 or 30 block sequences. Can someone double check? Is it -35, -30? And for translation, what's the starting signal? Bacteria. It's the shine gano. I probably didn't spell it right. Otherwise, spell shine gano. EALGARNO. Okay. external sequences. And then there's also the AUG. Right? So, the elongation for both of the when replication in a probably easier to look for the stop codon. For replication to stop, actually this part, once it's finished, it's going to stop. It doesn't stop doesn't seem to be a problem for replication. But what's the stop signal for transcription? There's an intrinsic stop. There's also a row-dependent termination. Right? So you can say a row-dependent signal terminator. For translation, it's a stop signal. The stop codon. Right? So this is much more needed. Stop codon. And I guess the more complicated part is the actuating process. So for the transcription to occur is 5' to 3', and there's also a proofreading part. For translation, how does the translation go from N terminal to C terminal, or C terminal to N terminal? What's the direction of translation?
N to C. N to C.
N to C? C to N? N to C? I remember N to C. Somehow they seem to agree. Yeah. So, and what's formation is also peptide bond. So, during the translation process, how many sites the ribosome has? It has a P site, A site, and E site. I probably should also add initiation factor here. Initiation factor here for translation. That's where the Shandaganno sequence is on. So the transfer RNA bring amino acid into the A site, the peptide bond join on the A site, and then move on to the E site, the release part in the E site. The process for replication is probably more. There's a lot of more things. You need a primer here, right? Primer. You need the polymerase. Oh, sorry. I should put proofreading in the DNA replication part. That actually, I'm not sure whether transcription has a proofreading. My recollection is that maybe we should double check it. Yeah, so proofreading here. But this one in eukaryotic will be different. It's going to have a post-transcription process. Okay, that's actually quite a lot for today. So let's summarize. Actually, a lot of biology. if you after the 4 years of college in biology, you can do this. This is really a rough summary. You probably can work on this in next class to work on it a much better way. So remember, this is just a bacteria, and we haven't talked about eukaryotic yet. So, okay, so tomorrow we are going to start a lab on sample microbiome in the environment, and some of you want to sample on your own, and you should really talk to me how you want. At least you can try. So, but the procedure is similar, except for water. If you want to see microbiome water, we need to look for some filters. I sent a notice to our lab instructor to ask for filters. And then respond, but we can probably borrow some from chemistry lab. So we just need to have a glass funnel and with filter paper, you can autoclave the whole thing so it's sterile, and then we just add various water. Say you can find out water from fountain here, or as a control, you can pull some water from the restrooms compared to sewage or small item. Yeah, so yeah, or you can buy a bottled water at the tap water. So and distilled water can be your control. So which one? I'm not. Okay, sorry. This is I think I go over 5 minutes now. Wait a minute, this class ended at 10. I still have to. I was actually trying to wrap it up. Yeah. Okay, so okay, I guess I can still finish the last slide. The last slide actually is when you saw a question in your GRE about the protein structure. So protein structure, secondary, tertiary, and correlate structure. The primary structure will be just sequences. The secondary structure is how the primary sequence form as the basic building block. The secondary structure will be just the two main secondary structure, beta sheet and alpha helix. And one of the key differences between beta sheet and helix is how the hydrogen bonds are formed. In beta sheet, the hydrogen bond is formed between strands. Those are the beta sheets. In alpha helix, the hydrogen bond is formed along the alpha helix. So this is probably we're actually going to do a lab on this. So how does this look like? It's basically like this. So this is beta sheet. And that's what you are looking at, those are the backbones of amino acid. So now, how does the hydrogen bond form here? Hydrogen bond form should be formed between the strands. So that's basically hydrogen bond formed between strands. So this is beta sheet. And for alpha helix, it's the okay, this not going the wrong. Alpha helix, the hydrogen bond, the green hydrogen bond is formed along the axis, and alpha helix is also right-handed. So if I turn this, you see the spiral direction is right-handed. So it's just like the DNA helix. So if I try to grab this alpha helix with my right hand, my thumb in the direction is moving forward. So this is right thread. It's basically the same thing with all the threading here. Right? So those should also be right-handed. If I grab with my right hand, this should go down. This is right hand. The same thing, if I grab this word, it should go down. So it goes down again. Either way, it's also right. It doesn't matter how I flip. So that's basically, and okay, that's basically it. Well, I already told you the alpha helix is the main bond is hydrogen bond around axis. So, here's another question. I'm going to skip it. You can just go to the summary. So, well, see, this chapter covers so many things. DNA double helix. It's also right-handed, and basically DNA secondary structure caused by inverted repeat. This also found in RNA. That's actually how transcription terminates. Right? So in intrinsic termination or role dependent also need that. How the melting temperature of nucleotide is estimated. Basically, coli genome, DNA replication, concept PCR, transcription, promoter, working on about minus 35 hours. So I was right there. Terminator and opera. Translation, you need the strand diagonal sequence. That's where the ribosome binds. And the translated part called operating frame. And tRNA has anticodon. Anticodon has Weibull position that allows one tRNA can recognize several sense codon on RNA. Protein structure has primary, secondary, tertiary, quaternary structure. And alpha helix and beta sheet are the two major secondary structures. And they are formed by hydrogen bonds. Alpha helix is within helix. Beta sheet is hydrogen bond between strands. For the factors to form protein structure will be hydrogen bond, hydrophobic interaction, electrostatic interaction, that's between positive and negative, bindable interaction. That's a very vague thing. I'm going to explain. If you think about all the interaction boils down, all goes back to that. But if you goes back to the quantum chemistry, quantum mechanics. But we actually conceptualize higher up, say something specific like hydrogen bond, hydrogen interaction. But boils down all to be that. Okay, so if you finish this chapter, that's a very good portion of MK.GIE will be covered. This is okay. Okay, I'll see you tomorrow. And some of you want to talk to me about the presentation of bonus point, how you want to do your own project. You want to do the lemon thing, right? Yes. Okay. Oh, by the way, so because of the snow, we are 2 weeks behind. So we're probably going to have another exam after we finish chapter 8. Not to leave us to end of March or early April for another exam. And then we have, according to the syllabus, we have third exam and final. But that means they will be basically scheduled as back to back. Do you want to have the third exam, or you want to just have the final? I'm going to leave to you. So,
third exam.
Supposedly cover all three. So if I were you, I would pick the third exam just to spread out the risk. It's also a practical round of final. Final basically combines everything again. But that just means we probably, since we're going to have the third exam around early April, and the final will be early May, that means not much time between basically we took the third exam, and then I gave you the grade, and then that's the final exam. You do your project. Okay, I'll let you. I'll let you think about it. Yeah, what?
I said, can we think about it?
Yeah. So.